Have you ever wondered what happens to an eDNA sample after you collect it from the environment? Let's take a journey behind the scenes and demystify the lab process that transforms eDNA into a biodiversity treasure map. Our adventure begins with collecting eDNA from various sources: water, soil, air, or even scat. In these environmental samples, DNA material comes in all shapes and sizes, within cells, tissue parts, or even as free-floating molecules. Next step, extraction. We thoroughly mix the sample by shaking it vigorously. Then we add chemical reagents to break open the cell membranes to release all the DNA molecules. Next step is purification. We add solutions that clean the DNA mixture by removing unwanted substances, leaving us with just the pure DNA. Now that we have that pure DNA, we use polymerase chain reaction, or PCR, to amplify it. PCR functions as a copy machine for DNA, creating many copies of specific DNA sections, making them easier to detect. It's like using a magnifying glass to reveal DNA traces even in tiny quantities. If we're interested in a specific organism, we use quantitative PCR. This technique can monitor the real-time presence and amount of a specific DNA sequence during the PCR amplification, similar to how it was used during the COVID pandemic for diagnostics. To explore multiple species, we use metabarcoding. This technique uses high-throughput sequencing machines to read the amplified DNA fragments and generate sequences, the genetic fingerprints of different organisms in a sample. The generated sequences are compared to a global database to identify which species they belong to, much like scanning a barcode at a supermarket to know what's in your shopping basket. And voila! You get a list of organisms detected in your sample. eDNA provides a powerful alternative way to discover biodiversity, aiding in nature research and conservation. Want to learn more about eDNA and its applications? Reach out to the Sequench team in New Zealand or Lithuania. Discover the hidden world of biodiversity with eDNA.